Right, when we finished off the notes, we were talking about fats or lipids. Okay, and I also talked about saturated and unsaturated. And again, this is where we ended up with the animal fats and the plants. Okay, plants are liquids at room temperature, and animal fats are solids. So, like butter, Crisco, things like that. And then olive oil and canola and all that are. So, it's better to use those because they're better for you. Okay, so why do we have fats? They store energy. Okay, if you are trying to lose weight, you'll notice that you have to exercise for a little bit longer when you have fats because your body will burn carbohydrates first. And then once you've been burning it long enough, then you'll hit the fats. Fats have much more energy in them, but it takes a lot more to get them started. I always think of like when you're building your campfire, you know how you have to get it started first with the little twigs? The little twigs are the carbohydrates. Okay, they burn up really fast, and that's what carbohydrates do. And then you add the bigger logs once that fire's going, and then you burn those, and they burn longer. The bigger logs are the fats. You need more fire to get them going, but once you get them going, they give you off more energy, and that's kind of how fats work, okay? So we save fats in our body as adipose tissue, okay? That's our food reserve. And like I said, we need some fat in our diets, we need some fat in our bodies, but most of us have kind of gone a little crazy with that. Sorry about that. Like I said, I'm number one on the list. Okay, adipose tissue cushions your vital organs and it insulates your body. So yes, you need some fat. Okay. All right. So another thing about fats is they make up steroids. Okay, they're carbon ring. They have four infused rings, and they have special, like cholesterol. You guys have heard of cholesterol before. Everybody thinks cholesterol is really bad for you. In large amounts, it is because, yes, it'll lead to hardening of your arteries and you'll have issues. But we do need cholesterol. It's a part of our animal cell membranes. If we don't have cholesterol, our plants, I mean, not our plants, our animal cells, our cells don't do the things that they're supposed to do. So we do need to have cholesterol. Okay. Too much cholesterol, and if you don't know, above 200 is supposed to be bad for you. If you don't know your cholesterol, you guys give blood, they'll test your cholesterol, and then you'll find out what you have. But anyway, if you have high cholesterol, it'll contribute to cardiovascular disease. In other words, it'll fill in your arteries and then you'll have stroke, heart attack, icky things like that. Okay. All right. Proteins are the next biomolecule. So we talked about carbohydrates and fats. Now, proteins, they account for 50% of your dry mass. Again, we got to take water out of there because 70% of us is water. If you take out the water, then of the what's left, the 30% that's left, half of that is proteins. Okay. There are some proteins that speed up chemical reactions. These would be enzymes. Their job is to control and make sure the reaction occurs. Okay. And then other proteins do a lot. We have a lot of different proteins. Okay. We have a few fats, we have a few lipids, we have some carbohydrates, but man, proteins is the champ. Okay. They help us with defense. In other words, they help blood cells fight off diseases. They store things for us. They transport things around and they communicate between the cells. They move things around. They're structural support. I mean, our muscles, our skin, our bones, these are all made out of proteins. Our brain organs, all our organs are made out of proteins. So we have a lot of proteins with a lot of different functions. Okay. Again, enzymes is a special protein and their job, they act like a catalyst. And what a catalyst does is it's in charge of controlling reactions. Now it says to speed up, they can actually slow down chemical reactions too. Okay. So, and the weird thing about it, like if you eat a cheeseburger, okay, you got the cheese, you got the meat, you got the bread, all you have an enzyme to break every single one of them down. There's not like a general enzyme that breaks down food. You have to have a different enzyme for every starch, every, protein, everything has to be. So we have lots and lots and lots of enzymes because they all have a different job to do. Okay. The nice thing about enzymes is they can do their job over and over and over and over. It's like you don't run out of them. They do their job and then they move. They're like factory workers that never take a break. Okay. They work and they work and they work and they work. All right. We have proteins. They're constructed, like we said, carbohydrates were made out of monosaccharides. Proteins are made out of amino acids. An amino acid we have in our body, we have 20 of them. And the sequence of the amino acids determines what protein you have. Okay. So we take a bunch of amino acids together and we attach them, and then we get our protein. Same thing, like I said, if you want to consider each Lego an amino acid, when you put a whole bunch of Legos together, you get a structure. When you put a whole bunch of amino acids together, you get a protein. Okay? And a protein, its function consists of whatever these polypeptides are. So the sequence of amino acids is really important. It has to be exact for the protein to work. If you change the sequence, you change the protein. Okay? And again, what it does is the amino acids determines the protein's three-dimensional structure. If you know people that have issues with, like, they're lactose intolerant, for instance, and there's a lot of Native Americans that are lactose intolerant. That's a very common disorder. What happens is they can't make the enzyme lactase. That part doesn't work anymore. And it worked when they were younger in most cases. But as you get older, it doesn't work anymore. So you've got this issue where you can't take in dairy products because you can't break them down. Okay, that'd be an example where the structure of lactase, the enzyme that breaks down lactose, milk sugar, has been changed. Okay, so the structure, the amino acid sequence determines the protein's three-dimensional structure, and it determines what it does, how it works. Okay, it binds somewhere, it sticks, it has like what they call, if it's an enzyme, it has what they call an active site. And it binds to it, and then it causes the reaction to occur. If it's like a key, nothing's going on, the enzyme comes, attaches to that, and it's like, oh, now it's time to get busy. Okay, so they bind to another molecule, and then things happen. Okay, there's when you're talking about a protein, they have 4 different levels of structure. Okay, the first one is just the primary structure, and what it is a sequence of amino acids. Okay, whatever, and again, there's 20 of them, and they're in your book if you want to read them. There's like alanine and phenylalanine and all that, whatever the order of those amino acids is. And not every protein has every amino acid. Okay, it's the sequence, so it might have alanine, phenylalanine, leucine, and then alanine. I mean, it's just whatever the structure is. Okay, and they can repeat, but they don't have to have all 20 amino acids in every protein. I probably should have said that sooner. Sorry. Okay, the secondary structure is one of 2 things. They either coil up, once they get the protein made, they either coil up or they fold up. Okay? And then there's the tertiary, which is where you get interactions. Okay? Among the side groups, there's groups on the side that are sticking out. And if they react with other things, that's their tertiary structure. And then finally, they have the 4 structure, the quaternary, and that's where they attach to other peptides. Okay, so, and I'll show you this better. Me explaining it sometimes isn't so great. I'll show you it in here in a little bit. Okay, and then the last biomolecule is, where are we at here? 8. Okay, I'm just checking our time. Is nucleic acids. Okay, and nucleic acids we've talked about a little bit already is DNA and RNA, and their job is to express a hereditary information. The crazy thing about what DNA is DNA is the sequence of amino acids. Remember, the sequence of amino acids determines what protein you have. So the information to do all that is in your DNA and into little units, and each unit is called a gene. Okay, so you have genes for your hair color, and genes for your eye color, and genes that you have a mouth, and you have teeth, and you have ears. All that's in your DNA, and it's a sequence of proteins. Okay, so genes consist of DNA. So if you take DNA and break it up into little pieces, you have genes. Okay, and a nucleic acid is also made out of little units called nucleotides. And a nucleotide is actually just 3 things. It's a sugar. In the case of DNA, it's deoxyribose. And it's a phosphate group, PO4, and 1 nitrogen base. And there's 4 nitrogen bases in DNA. So again, it's kind of like a nucleotide is 1 Lego, and then you put a whole bunch of them together, you get a big long strand of DNA. Okay? So there are 2 types of DNA. 2 types of nucleic acids, sorry about that. There's DNA and there's RNA. Okay? DNA is deoxyribose nucleic acid, and RNA is ribonucleic acid. And what it is there's some differences between the 2. One of them you can see right away. The sugar in DNA is deoxyribose. Ribose, excuse me. And in RNA, it's just ribose. So they have a different sugar, and it's off by like 1 carbon, I think. Not a big difference, but okay. DNA replicates itself every time a cell divides. Okay, it provides in the DNA, it actually has instructions on how to replicate itself. Okay, DNA is the ultimate boss. It controls everything about you. It's the blueprint to build you, and it actually takes care of itself as far as making more. Because every time a cell divides, you got to make sure you have the same DNA in each one. So DNA takes care of that itself. And then what it does is if you need a protein, let's say you get a cut and you need to repair the skin cells, what will happen is DNA will open up the gene for making more skin cells, and then RNA, messenger RNA, will go in there and make a copy of it. And then the DNA will close back up again. So it uses the blueprint, but it's still there to use again. The messenger RNA will leave the nucleus because that's where you find DNA. DNA stays in the nucleus. That's its office, and it never leaves its office, okay? But the messenger RNA leaves the office, and then it goes out into the cell, and it makes these proteins, okay? And that's called gene expression. So the DNA controls it, but really the RNA is the one that does all the work. The RNA builds the proteins. The DNA is the information to build it, okay? So when you're looking at nucleic acids, again, they're made out of many, 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 many nucleotides, okay? And a nucleotide is just a small unit with a sugar, a phosphate, and one of the nitrogen bases. And DNA has 4, okay? So when you're talking about these nucleotides, the pentose sugar, that would be the ribose or deoxyribose, depending on if you DNA and RNA. And then you have one or more. You have your DNA, a base, and the phosphates, okay? I don't know that you need to know the nucleoside, but nucleotide is what you're looking for. That's the monomer that makes up DNA, okay? So a nucleotide is a nitrogen base, a sugar, and 1 phosphate base. There's 2 groups of nitrogen bases. There's 4 of them, but each one is in its classification. There's the pyrimidines and the purines. The pyrimidines are cytosine, thymine, and uracil. There's a different, in RNA, it doesn't have thymine. It has uracil. So DNA has thymine, RNA has uracil. That's why there's 3 pyrimidines. These are a single ring, okay? A purine is adenine and guanine, and they're a 6-membered fused ring, so they're double. So what happens is a pyrimidine and a purine are always together. Cytosine always goes with guanine. C goes with G, always. A goes with T, always. You never change that, and that's why when DNA makes a copy of it, it follows that code, and it never changes from that code, and then it'll make exact copies of itself, okay? So again, and I said that before, in DNA, the sugar is deoxyribose. In RNA, the sugar is ribose. That's one difference between them, okay? And this is the structure. There's an animation here. I will put this slide on, then you can go through it, but I'm not going to do that because we don't have much time. Okay, so the DNA is called a double helix. It's like a spiral staircase, okay? The steps of this case are the nitrogen bases. There's 2 of them stuck together. And the sides of your ladder are the sugar and the phosphate. And the bases always go forward from the sugar. They're connected to the sugar, okay? One DNA molecule has many, 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 many genes. Think about all the characteristics of us. And they're all in that information, okay? And again, I said this already. The bases form hydrogen bonds between the adenine and the thymine and the guanine and the cytosine. So A and T always go together. G and C always go together. That code never changes, okay? And that's complementary base pairing. You cannot change that. If you change that, then that organism is going to die, okay? I know that sounds weird, but you're going to have different proteins. So the feature of DNA, that's why it can make identical copies of each other, because it always pairs in the same order, okay? RNA is a little bit different. RNA is single-stranded, and it has a complementary basing too. C and G always go together, but now A and T don't go together. RNA doesn't have thymine, so it has uracil. So A and U go together, okay? That's a difference between them. And DNA is a double helix. RNA is just a single strand. And there's more forms of RNA. There's 3 forms of RNA. And that's it. I made it. Okay, so we're done with the notes for this chapter, and we'll talk more about review and everything, but now we're going to take our first test.